Hello, everybody, and welcome to our lecture on genetics. So in this lecture, we'll be looking at some of the, just the very basics in genetics, and we'll be looking at very basic inheritance. We're not going to go into, delve into a bunch of hyper detail about the inner workings of genetics, but we'll learn a little bit about the basics and how it kind of relates to evolutionary traits. And genetics is everywhere, right? And DNA is everywhere. We find DNA in things like pollen grains. We find DNA in our cells, like these little somatic cells here. We'll even talk a little bit about something called apoptosis, which is the programmed death of cells, as well as talking about DNA, the DNA molecule, and chromosomes. So to give you just some basic background information, we should all kind of remember this from either our kind of basic college biology class or our high school biology. But there are two types of cellular organisms on this planet. You have prokaryotic cellular organisms, and the way we kind of tell the difference between the two is, most times prokaryotic organisms are single-celled, and their DNA is not contained within a membrane-bound nucleus, right? Eukaryotic organisms, on the other hand, are multicellular organisms, things like your cats, your dogs, your humans. There are even some single-celled eukaryotes as well. But this, it's defined by having its DNA contained within a membrane-bound nucleus. So that's the big difference between the two, as well as some other differences. But that, for our intensive purposes, that's the difference we need to be concerned with. So your prokaryotic organism is a single-celled organism without a nucleus. It's the earliest form of life on Earth, arriving at 3.5 billion years ago. Your eukaryotic organisms, on the other hand, arose at about 1.2 billion years ago, and some single-celled eukaryotes, such as yeast, are, eukaryotes kind of a little bit of flexibility, whereas prokaryotic organisms are generally single-celled. We do have both multicellular and single-celled eukaryotic organisms. And we're not going to go into all the details about all the different apparati on the inside. We'll talk a little bit about mitochondria. We'll talk about DNA and a little bit of protein construction, but that's about it. So eukaryotic organism is an organism whose cells have a nuclei, right? That's very important. You will be tested on that. In eukaryotic organisms, or your multicellular organisms, there are two different types of cells, or two kind of categories of cells. We have somatic cells and gametes, right? Your somatic cells are the cells that form most parts of an organism's body, like these skin cells you see featured on the left. Or you could have gametes, which are sex cells. And these are your sperms, your eggs, or if we're looking at plants, it would be the pollen and the ovules. So just showing you where the ovules are in the plants, and these are what contains the pollen, which in essence, once it is kind of completed, the pollen, those ovules open up, the pollen begins to be spread, and that is how sexual reproduction in plants occurs, right, through pollination. This is just showing you neurons, so even the motor neuron cells within your brain are, and throughout your body are somatic cells as well. So basically, if we're talking about a sex cell, it's a gamete. Everything else is going to be a somatic cell. So if we look at the DNA molecule, we're going to look at your nuclear DNA, which is contained within the nucleus of a cell. It's what makes up the chromosomes. Each DNA molecule equals one chromosome. And the complete set of the chromosomes is called your genome, right? And your genome is homoplasmic, which means that it is the same in every cell. And then on top of your genome and on top of your chromosomes and your DNA, you have a set of regulatory switches that essentially wrap around your DNA that we refer to as your epigenome. When we look at chromosomes and chromosome counts, you have two different ones. You have your diploid number, which occurs in your somatic cells, right? This is the full chromosome count in each nucleus of the cell. It is a full set of your DNA. Your haploid number, on the other hand, that is the number of chromosomes carried by each gamete, right? It is a half set, right? Because remember, if you're contributing a sex cell in reproduction, and that's what goes to create an organism, right? If we're speaking in terms of humans, you're providing half of the DNA for that new organism in your sex cell, and your partner is providing the other half of that DNA in their sex cell. And there are different numbers of chromosomes in different species, right? For example, chimpanzees or orangutans have 48 chromosomes. Apples have 34. Algae, look at how many chromosomes that algae has, 148. Algae is actually fairly adaptable and can live in a variety of different environments. Salamanders, 24. Houseflies, 12. Camels, 70 chromosomes. Potatoes, 48 chromosomes, right? So orangutans and potatoes have the same amount of chromosomes, right? So it just shows you in the variety of life that we have on this planet, there is kind of some slight differences in terms of chromosomal DNA. This is showing you the human karyotype, which consists of 46 chromosomes of various sizes in 23 pairs. Showing you a close-up image of a chromosome there. This is a kind of a pip chart of chromosomes as well. You don't really need to know too much about this, but we're going to look at number 9 when it has the ABO blood group. We're actually going to spend a bit of time talking about the ABO blood group. This, a little bit later in this lecture as well as in future lectures. We are also going to look at the hemoglobin beta chain. Which is very important in the production of proteins as well as the production of your blood cells. We're going to look at a specific disease that affects the hemoglobin beta chain in terms of blood cell production, a disease called malaria. To look at the DNA molecule, we already talked about your kind of nuclear DNA a little bit, but you have your mitochondrial DNA as well. It's contained in the organelle within the cell cytoplasma. It's, of course, inherited from your mother and travels along the female line of your family. And this is why we can use mitochondrial DNA to kind of trace ancestry. Your mitochondria, remember, is from your biology classes. It's responsible for converting sugars and fats into ATP, which is an energy source for the cell. And a single human liver cell, to give you an example, has between 1,000 to 2,000 mitochondria, which makes a lot of sense because your liver is a very high-functioning organ within your body. It requires a lot of energy to run. It's essentially part of your blood filtration system. So it requires a lot of mitochondria. It requires a lot of ATP. So those levels of mitochondria will vary depending on the function of the cells in which they exist. So the DNA molecule is our blueprint for life. It's a chemical template for every aspect of an organism. It has a double helix ladder-like structure. The ladder verticals are the phosphates and sugars, and the latter rungs are made up of 4 types of bases. You have adenine, thymine, guanine, and cytosine. They come in complementary pairs. When you find an adenine or an adenine, you should find a thymine. When you find a cytosine, it should be paired with a guanine. Right? When this does not happen, you have a mutation and something is amiss. This is just showing you a visual representation of the double helix-like structure. Remember, it's the phosphate-sugar backbone, and in between that phosphate-sugar backbone, ascending in a spiral is your complementary base pairs. So a phosphate plus a sugar plus one of those nitrogen bases equals one nucleotide. And there is a soup of these in the nucleus as well as in the cytoplasm of the cell. And that nitrogen base can be either an adenine, guanine, cytosine, or a thymine. DNA is a self-replicating molecule. It's part of the cell division, both meiosis and mitosis. And this is a very easy way to determine between the two. If we're looking at sex cells, we're looking at meiosis. If we are looking at somatic cells, we are looking at mitosis. DNA makes identical copies of itself, also as well as chromosome types, and it occurs in homologous matching pairs, right? It's one in each pair from each parent. In terms of chromosomes, you have, much like your cells, you have two different categories of chromosomes. You have your autosomes, which are the non-sex chromosomes, and then you have your sex chromosomes, which we're all kind of familiar with. That's your X and your Y chromosome. Females carry only X chromosomes, while males have an X and a Y chromosome. But this is not exactly necessarily the truth. As a matter of fact, females do inherit that Y chromosome. It just becomes deactivated, and we'll talk about how that happens. And the father determines the sex of the offspring in humans, right? So, and it's kind of interesting, you know, if you think about back in history, all of the kind of royal families in which women were kind of shunned or divorced, or kind of in some cases even beheaded for not producing a male heir, when in fact it's actually the male of our species that determines the sex. So if we look at mitosis real quick, it's the production of identical somatic cells. It's DNA replication followed by one cell division. It happens in a diploid cell, which is a cell containing, remember, a full set of your chromosomes. If we look at another form of mitosis called rapid mitosis, is what is actually responsible for the growth of an individual from one cell to a baby in just 5 months. This is, during embryonic development, this is the fastest in which your body is going to grow for the remainder of your existence, right? That's why this is termed rapid mitosis. If we look at the other form of cell division, meiosis, which is the production of gametes, you have one DNA replication followed by two cell divisions, right? So then you end up having a half set of the DNA, right? Remember, your gametes are haploids, which are a half number of the chromosomes in your DNA, and do not result in identical cell copies. Errors can occur during meiosis. You have non-disjunction and translocation. We'll talk about both of those. To determine sex in males, you have the Y chromosome, which carries the SRY gene, which is especially important for sex determination. It turns the gonad into a testes and stimulates the SOX9 gene. The SOX9 gene produces a hormone called Müllerian-inhibiting substance, which makes the female tubular reproductive structures regress. And yes, you are essentially hearing and reading that correctly. We are all essentially, in our embryonic development, we all start out having both elements of both kind of male and female sexual organs. It's when this kind of Y chromosome in this SRY gene becomes activated and stimulates the SOX9. This is when it begins to cause female reproductive structures to regress, and that's when you have sex determination occurring for males. If there's a mutation in that SRY, it fails to switch on the SOX9 gene. You have no SOX9 gene in an XY fetus. You have a baby that is born looking female because female structures persist, but you also have gonadal digenesis. You will still have testes actually inside of the body. So it's how we can get individuals who are born kind of with more masculine features, or what we would consider more masculine features, and we have individuals that are born with more feminine features, and they still have their kind of, one or the other sexual organs or sex determination. And another kind of element is, and it's kind of important to mention, that there are actually 23 different categories of intersex where you could be born with kind of elements of both the male and female anatomy. It really just depends on the interaction of your DNA and that Y chromosome with the SRY and SOX9 genes. So sometimes your DNA does not replicate properly. We talked about this or mentioned it in an earlier slide. You have translocations, which is a switching of entire chromosomal sections, and then you have non-disjunction, in which the DNA fails to split in some areas, right? And we're going to talk about how, a very small percentage of our DNA actually codes for the production of protein. So we have a lot of what we would call redundant DNA, or what used to be called junk DNA, floating around in our genome. And that is not necessarily junk DNA. That DNA serves a significant purpose. When we have essentially non-disjunctions where the DNA fails to split, that junk DNA can kind of come in and repair certain sections of the chromosome. So when DNA does not fix itself with our kind of natural DNA fixing mechanisms, you have germline mutations. These are hereditary mutations which are passed from parent to offspring and account for roughly 5 to 20% of all cancers that we see. An example would be the BRCA1 gene mutation, which leads to hereditary breast cancer, because the BRCA1 and 2 genes are genes which repair DNA and suppress cancer growth. So if you have kind of an issue with either one of those genes, or a mutation happens in either one of those genes, it reduces your ability to repair DNA overall, and it also reduces or increases your chances of developing breast cancer. You also have other repair mechanisms that are kind of currently being investigated. This notion that the length of your telomeres has something to do with your ability to repair DNA and your ability to essentially live a long lifetime. Some of the longest living creatures on the planet, like tortoises, have very, very long telomere lengths, and this is kind of the end section of your chromosome. And when your cell division occurs and when the DNA is splitting and there is a error, a tiny piece of that chromosome gets shaved off on the end, and that DNA is then used to repair the DNA which fails to split properly. So talking a little bit about Mendel's kind of contributions to genetic theory, of course, we have the law of independent assortment, which says that hair color, for example, is inherited independently from eye color. So what this is really saying is that different traits within your body are inherited independently from one another. So if we look at Mendel's original work in terms of pea colors, you have the pod color, which is inherited independently from the pea color. In terms of genes, you have two basic categories of genes. You have the structural versus regulatory genes. Structural genes are responsible for the production of body structures, whereas regulatory genes turn other genes on and off, or act on some of these structural genes. These are also known as homeotic or Hox genes, as well as master genes if you're reading some of the older literature. And essentially what these genes do is they act as regulators, right? They turn the gene on and tell it to send a signal out. An example that we like to use is the D12 and D13 genes within humans, or actually within any mammalian that has limbs. And those are actually responsible for the construction of your arms and your hands and your fingers. If we upregulate that D13 gene, you could actually, keep increasing the signal to it. We can actually increase the number of fingers an individual will develop on one hand. If we take that same gene and we start to prematurely cut the signal off from it and turn it down, then we'll have an individual that would be born with less than the normal amount of fingers. So let's talk a little bit about proteins, and proteins are chemicals that make up tissues. They also regulate functions like repair of cells and growth of your tissues. Proteins are made up of amino acids, and there are 20 different types of amino acids, and some protein diseases can be pretty hefty. The one you're kind of seeing here in the corner here, I know it looks like the individual is starved. Actually, they are not. They have a protein production disorder in which they cannot produce enough muscle tissue. So it essentially, depending, no matter how much they eat, it consistently looks like they're wasting away. Structural proteins are responsible for the physical characteristics of your tissues, whereas regulatory proteins, much like kind of your structural versus regulatory genes, are responsible for functions, things like enzymes, hormones, and antibodies. Protein synthesis, or the production of proteins, involves two different steps. You have transcription, which is the unzipping of the template for the RNA, and then you have translation, which in the template attaches to ribosomes, begins to become twisted and form into a protein. If we look at producing proteins, just as I kind of mentioned a little bit earlier, DNA, there's a tiny, tiny percent, only 1 to 2% of your genes code for the production of proteins. About 45% of your DNA is unique non-coding genes, right? Another less than 1% are pseudogenes. We won't really worry about those. And then you have 50% of your DNA is roughly repetitive sequences, right? This is what will be used in your DNA repair mechanisms. So most of your DNA, in fact, is non-coding. So what is so special about proteins, right? Well, there are many different kinds of proteins. I'm not going to expect you to know them all or, the specific types, but these are just some examples. Some structural proteins like collagen and keratin. You have transport proteins like hemoglobin in your blood. You have immune system proteins like antibodies. You have contractile proteins like actin and myosin, which work to move your muscles and help extend and contract your muscles. Some hormones are proteins as well, like insulin and growth hormone. Some pigments are proteins, like the rhodopsin pigment in your eyes. Enzymes are also proteins, like lactase and lipase, right? Lactase you may be familiar with because that's what's required to digest lactose, right? So if you're lactose intolerant, you do not produce the protein lactase, therefore you are unable to consume dairy products. And on each of your cells and how these kind of proteins work within your body is that there are channels and receptors on cell membranes, which are also proteins, and these kind of act as gateways into the cell and are kind of a really specific lock and key system, right? Only a specific protein will fit in its receptor, right? No other protein will fit in there. This is showing you just kind of how complex proteins can become. This is the CAMK2A protein. It's related to the disease ischemia, which means inadequate blood flow to an organ, which commonly affects the heart tissues, right? So if you produce what this is called as an abhorrent protein, if you are an individual who has a specific mutation that produces this CAMK2A protein, you have a higher likelihood of developing ischemia to the heart. In all life forms, the same 61 DNA codons specify for the production of the same 20 amino acids. Those amino acids are then used for the production of proteins. This is just showing you protein synthesis. I suggest that you kind of look at this chart in your textbook. I'm not going to test you on all the different steps, but it is important to kind of know how a protein is made. In essence, a codon, there are three bases that specify a particular amino acid. That amino acid is then brought to the ribosome. There, the amino acids are bonded together, forming a chain. The chain becomes twisted and folded and combined into a complicated 3D structure that is a protein. And their unique exact shapes are very important for their proper function in the cells. So let's look at some differences between DNA and RNA. The four bases in DNA are cytosine, guanine, adenine, and thymine. The four bases in RNA are cytosine, guanine, adenine, and uracil. Their nucleotides with these various bases are floating around in the nucleus. Another big difference is DNA is double-stranded, whereas RNA is single-stranded, and the sugar molecules for each are different, right? In DNA, it's deoxyribose as the sugar molecule. In RNA, it is simply ribose. There are some diseases that we tend to see in prevalence within human populations today that relate to protein production. We have one in which that's called Marfan Syndrome, in which the individual has too much connective tissue and they have a hard time with muscle deposition. And these individuals can kind of be identified in terms they're very tall, they're very lanky, very thin, and they have exceptionally long hands and fingers. And we actually have a historical figure whom scientists today believe may have had Marfan syndrome. Unfortunately, they will not let us dig him up to look at the body and do the DNA testing. When looking at protein synthesis, the whole gene gets transcribed, but only certain sections of the gene actually code for the protein. These are called exons. Some sections of the gene do not code for the protein. They don't code for anything, and those are called introns. During the mRNA transcription, essentially your transcript must be edited. The non-coding parts must be cut out. So in essence, the exons are the ones that code, get to leave the nucleus and get translated into a protein, and the introns are cut away and they stay within the nucleus, right? This is where you get that kind of soup floating around of introns. So a real good way to say it, to kind of remember this, or a good way to remember the difference between the two, is exons get to exit the nucleus, introns stay in the nucleus. So introns for in, exons for exit. And introns account for a large portion of the DNA, right? Remember, about 98% of your DNA is non-coding. So let's take a quick look at polymorphisms, genotypes, and phenotypes, which is essentially genes and their physical expression. If we look at chemically identical alleles, they are termed homozygous, right, if we're looking at a specific trait. If you have chemically different alleles, these are termed heterozygous, right? A dominant allele within a allele pair is always going to be expressed. For a recessive allele to be expressed, you must inherit a copy from each parent. Each gene has a specific physical location, otherwise known as the locus, and loci are valuable to understanding what we can consider genetic variation. And alleles on different loci are chemically alternate versions of the same gene. So when we're going to look at polymorphisms, and we're going to talk about a very, very important polymorphism, and these are essentially kind of variations on DNA that still are functional and still work. We're going to look at a polymorphism in the hemoglobin beta chain. So some genes have one allele while others have more. And remember Mendel's law of segregation, which is a parent passes one allele to each offspring. And this is where we kind of end up with having siblings that have albinism, right? If we look at albinism as a double recessive trait, which is what it is, 1 out of 4 children are going to, if you have 2 heterozygotes that are married, and reproduce, 1 out of 4 of those children are going to be born as an albino, right? Because 1 out of 4 of those expressions is going to have that double recessive trait. The ABO blood group, for example, or ABO blood group system, each of us has 2 alleles for this trait, right? From one parent, you may inherit an ABO. From the other parent, you will inherit an ABO, right? Thus, the genotypes for this trait are your blood types, your AO, your AA, your BO, your BB, your AB, and your OO, right? In the ABO blood system, A and B are each dominant over O. So what you see expressed in the person, the phenotype, in essence their type, is due to the dominant recessive nature of alleles. And that's how it works with pretty much any of these simple inheritance alleles, right? And it's very important to kind of understand the difference between a genotype and a phenotype, right? The genotype are the genes that you inherit. The phenotype is how those genes are expressed. Another way you can kind of think of this is the genotype is all the genes within your body, and the phenotype is how you kind of look like on the outside, all of the different expression of those traits in your physical appearance. So going back to blood, if you have type A blood, that means your genotype is either AA or AO. If you have type B blood, that means your genotype is either BB or BO. If the A and B alleles are both expressed, right, if they end up in the same person, you will have type AB blood if your genotype is type AB. If you have type O blood, that means you have the double recessive expression, which means you received 2 O alleles. So this is showing you the process of agglutination. So in essence, what happens if you are transfused with the wrong blood type while you're at the hospital, the antibodies on your and the antigens on your blood cells will react. They will begin to clump together, and they will begin this process called agglutination, which essentially turns your blood into almost like a jelly-like substance. It's not a very good process. On the first transfusion, you may actually be okay if they address it and kind of get to it quickly and deal with it. But if you are ever kind of transfused again with the wrong blood type, most likely you would not survive the process. So let's look at polygenic variation, otherwise known as pleiotropy. Much of our genetics today is based on the one gene, one protein model. In essence, one gene produces one protein, which has some sort of effect in your body. If we look at pleiotropy, on the other hand, this is the concept that a single gene can control multiple proteins and multiple different areas, right? If we look at one, if you have a mutation in the beta globin, then you will end up having a pleiotropic effect, which affects your eyes, your liver, and your heart, which increases chances for heart attack. It could cause liver failure, and it could cause blindness. So many traits are in our body and in our genes are polygenic, right, and are determined by genes at more than one locus, right? So that's why it's kind of hard to determine the root causes of genetic diseases, right? Let's look at an example. One thing we may think is pleiotropic as well as polygenic is autism, right? Yes, there are certain environmental factors that could cause a kind of version of autism, but it's not true blue autism. True blue autism is generally a genetic disease. It's less of a developmental disease, but it is a polygenic or it's a disease that's a pleiotropic effect of a polygenic trait, right? So it's really hard to kind of pinpoint exactly which genes are responsible for causing the disorder. All right, let's review real quick some Punnett squares and Mendelian genetics because you will be dealing with this in your lab activities for our genetics week. So to set the stage, right, are we kind of diploid or haploid organisms, right? Remember that these questions are kind of important as we move through genetics week. Are we a eukaryotic or a prokaryotic organism? How many alleles for a single gene do we inherit from each parent? So in essence, if you're following along and answering these questions, we are diploid organisms, we are eukaryotic, and we inherit one allele from each parent. So reviewing our kind of Punnett square, just setting it up, this would be the different kind of genotypes that would be the result of this particular expression, right? On the left-hand column, we have basically the genotype that was submitted by one parent, and on the top area, we have the top header, we have the genotype that was contributed from the other parent, right? So we're essentially seeing two individuals' DNAs coming together here for a single trait. So this is just showing you the different genotype expressions that we will have if 4 offspring were essentially made. So remember, dominant alleles may not always be in the highest frequency within population, but dominant alleles will always be expressed when we're looking at a simple inheritance trait. If we look at pedigree charts, these are generally used to trace genetic diseases or traits cross-generationally. They're very similar to kinship charts, if you know, or ancestry, or essentially, family trees. All of those things are kind of heuristic devices which do essentially the same thing, tracing something throughout a temporal time period. Usually it's based off of the expression of a dominant alleles, but not always. You could be trying to trace a double recessive condition that occurs in the family as well. So if we look at a pedigree chart, let's look at cystic fibrosis. A lowercase c, a green C represents a cystic fibrosis allele. A purple C represents a normal allele, or a capital C. If we look at the father and mother, these are, of course, 2 heterozygotes, right? They contain 1 copy each of the allele, so each parent has 1 normal allele and 1 affected allele. So if we're looking at the kind of when they have 4 children, you're going to have 1 that's going to be born with both normal alleles. They will have be completely unaffected. You will have 1 that will you'll have 2 that will be born a cystic fibrosis carrier, right? So they may generally be unaffected by the disease, but their children will have an increased likelihood of contracting the illness. There will also be 1 out of the 4 children will be born with cystic fibrosis. So in terms of genes and evolution, kind of bringing together what we learned in the last lecture to what we're talking about here, genes are not the unit of evolution that we are concerned about. The smallest unit of evolution, and if we are talking about evolution, is a single population. So natural selection acts on genes, whereas evolution acts on populations, right? So if we're referring to individual genes or the movement of genes from one generation to the next, we are referring to natural selection, right? If we're looking at how populations change over vast periods of time, like the development of bipedality or the increase in human brain size over time, this would be known as evolution, which acts on entire populations. So let's just finish up with a few concepts. We have Punnett squares that we'll review a little bit more of, exon activation in terms of sex determination, as well as some more pedigree charts. Remember, in terms of Mendelian traits, they are traits of simple inheritance, one locus and one chromosome. Remember, your chromosomes in current pair, thus two genes or alleles govern a Mendelian trait. A gene is, of course, a sequence of DNA bases, a code for a protein or part of a protein. An allele is an alternate form of a gene, and a polymorphism refers to the presence of alternate forms of a single gene, right, which will variably alter slightly the phenotypic expression of that trait. If we look at different modes of Mendelian inheritance, you have, of course, autosomal dominant, autosomal recessive, which we kind of talked about in terms of looking at cystic fibrosis, right? That's looking at autosomal dominance and recessiveness. And then there are sex-linked inheritance, right? And remember, chromosomes 1 through 22 are your autosomes, and your 23rd pair are your sex chromosomes. Autosomal dominant traits are coded for by one locus on one chromosome. The locus is on one of the 22 autosomes. Only one dominant allele is needed to express the trait, and it occurs in males and females equally. If we look at one autosomal dominant condition that we see, we have familial hypercholesteremia, which just means more presence of cholesterol in the blood, which later leads to heart problems in these individuals. But if we look at these alleles, right, if we look at the two lowercase c's, which would be the genotype homozygous recessive, you would have the normal genotype, right? If we have a heterozygous condition where you have one dominant allele and one recessive allele, you're going to have hypocholesterolemia or have a higher degree of chance of developing hypercholesteremia. If we look at, of course, the homozygous dominant condition, you most assuredly will develop hypercholesteremia. So if we look at another example or put that into a Punnett square, right, we want to look at what percentage of the offspring in this particular case would be born with hypercholesteremia, right? 50% would most likely develop it. If we look at an autosomal recessive trait, these are coded for by one locus on one chromosome. The locus, of course, is on one of your autosomes, and you must have both of the recessive alleles to express the trait, and it occurs in males and females equally. If we look at an autosomal recessive condition, we have albinism, which we briefly talked about already a little bit. If you have the homozygous recessive condition, of course, you're going to be born with albinism because you have both copies of the recessive allele. If you're heterozygous or homozygous dominant, inheriting two of the dominant alleles or only one of the dominant alleles, you are going to be born with a normal skin pigmentation and lack albinism. If two heterozygotes with this genotype kind of mate, what percentage of the offspring would be albino? 25%, right? Using the same Punnett square, what percentage of the offspring would be homozygotes? If you guess 50%, you are correct, right? So let's take a quick look at sex-linked traits. They are coded for by one locus on one chromosome. The locus is on one of the sex chromosomes, either the X or the Y. Females have two alleles for X-linked trait. Males only have one allele for X-linked traits and thus are termed hemizygous for their sex-linked traits. X-linked traits occur in males most often. If we look at what one sex-linked or X-linked trait, we have red-green colorblindness. These are the genotype of the alleles. The capital N is normal, whereas the lowercase n would be red-green colorblindness. So if you have kind of the genotype in terms of normal, you'll have a normal female. Then you can have a normal female carrier. If you receive a double recessive, you'll have a colorblind female. Then, of course, you can have a normal male and a colorblind male, right? So you can see how, in terms of females, they would have to kind of inherit both copies and have both copies be the issue in order for them to develop the condition, but males, on the other hand, only have to have one copy. So you can kind of see if a female is a carrier and she has a boy, that boy is almost going to be assuredly colorblind, right? So if a normal male mates with a carrier female, what percentage of the offspring will be colorblind or subjected to colorblindness? 25%. What percentage of male offspring are colorblind using the same Punnett Square? 50%. So how do males survive with only one X chromosome? Well, actually females too. In every female cell or every cell in the female body, one of the X chromosomes is deactivated. The deactivated X chromosome is also known as a bar body. You can actually put their cells under a microscope and see this kind of little black blob on the kind of outer edge of the cell, and that is the bar body. That's the deactivated X chromosome. So in terms of explaining X inactivation, during the first meiotic cell division, the second X chromosome in females is contorted into a shape that renders it inactive, right? This occurs within the heterochromatin. So this is just showing you the chromatin, the nuclear envelope, the nucleus, as well as showing you how these bar bodies are kind of deposited after that first division. And with that, we kind of conclude our kind of brief overview of genetics. We'll kind of move on to something we call population genetics in the next lecture, which will kind of tie these concepts into how we refer to genetics and evolutionary theory.